use it. Okay? That's the reason some of the bacteria can use amino acid, because they have the enzyme called deaminase. Once they have the deaminase, this amino group gone, this become phenylpyruvic acids. Very easy, let them go to next step is pyruvate. This guy can go to fermentation. And this guy can also go to TCA cycle. Go whatever they like. Let them can survive. So this is also an example, tells you bacteria can use lots of other energy resources in addition to glucose. Now the question back is, how do we know it's phenylalanine test positive? So let's say bacteria has deaminase. Take out of this amino base. You have this phenylpyruvic acids. How do you know it is positive? Okay, you have a phenylalanine test tube. You do a strict plating. What we're going to do? We will add in a 1 drop of FeCl2, ferric chloride. If the ferric chloride turns dark green color, it is phenylalanine test positive. Okay, so this is the second test. It's very easy. This is a very easy test. You get one slants, which is called the phenylalanine slants. We already have it. You're going to label them. Then you do streak inoculation. If bacteria use phenylalanine, we're going to add 1 drop of the we're going to add 1 drop of the ferric chloride. Just put on the top a couple of drops. You will see it turns dark green color, which means it's phenylalanine test positive. Okay, but we will test the next 2 weeks from today, just to give you an example. Okay, so this is the second test. The third test we will do is called Imvc test, or we say MRVP test. That will be the last test we use. So third test, we call it MRVP test. Okay, before I talk about this, we want to introduce you one group of biochemistry tests. That is important, called Imvc test. What are they? I means Indo. M is Meso Red. V is VP, Vocus Preskia. C is citrate. So for this test, we did here for SIM test. Meso Red and MV we will do right now. Citrate test we're not going to do. Why this test is important? This test is used to, in the food industry, used to differentiate E. coli and Enterobacter. Because the results is very, very funny. For E. coli, is Indo positive, Meso Red positive, Vocus Preskia negative, and citrate negative. For Enterobacter, Indo negative, Meso Red negative, Vocus Preskia positive, and citrate positive. Looks like this guy. Positive, positive, negative, 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 positive. This is like a sticky ends of the DNA. So that's why this test is important in the food industry, because they can differentiate E. coli and the Enterobacter. So we will do Indo for SIM test. We're not going to do citrate. Here we're going to do MRVP. So what is MR test? This is called a Meso Red test. Now make sure you understand that this M is not motility. This M is motility. Okay, SIM test is M is motility. Once you have MRVP, this M, IMVC, this M is Meso Red. So what is Meso Red? This is the fermentation products. Lots of the bacteria going through fermentation products will generate a very streak acid end products. and with gas or carbon dioxide, for example. Therefore, they will generate Meso Red. So Meso Red, which means we can test the bacteria going through fermentation. Their end products is citric acid end products. Okay, so how we do it? The testing is very simple. We will have these made in the tube. So simple it is. You shake it, you add the bacteria there. Okay, then you inoculate the bacteria there. This is about 2 ml. Inoculate, add incubation. Now how we do it? Next 2 weeks from today, you have a new tube. You add 1 ml there. You transfer 1 ml goes there. Then you add Meso Red reagent. If it turns pink, which means Meso Red Test positive. So very easy, which means the bacteria has final products, which is acid, citric acid products. So you understand here, you just see the E. coli is yellow color and the carbon dioxide generated. That's why it's Meso Red positive. Enterobacter is the other one. Okay? Now what is V? P means? That's called Vocus Pruskia. You can read it. I'm not going to write the full name. What is the VP going to test it? This means testing fermentation. Instead of you have acid and the products, you have a neutral and the products. And this neutral and the products usually showing up after another 24 hours. Okay? So in this reagent, we remove 1 ml into a new tube, and then the left 1 ml, we're going to add in VP reagent. If it turns pink color, it is positive. This will be doing this confirmation 2 weeks. And another thing is that for the VP, you may not see it immediately. So the tube you're going to put on your tray or in your rack for another week, we may show it pink color. Okay, so that's what I'm telling you. That's a little bit complicated. So VP, which is we testing, it's end products, which is neutral end product instead of its acid end products. Okay, so this is a third test. Okay, last test. So we finished this one. We're gonna talk about is this multi-channel. Okay, I'll just give you an example how we do it. So when we talk about all these bacteria, we talk from beginning to right now. Is we are actually talking. Do you have gloves? Yeah. We are talking about each individual testing, including 3 tube fermentation, oxidase, catalase, SIM, so many. But in the real life, when you're testing a bacteria, you're not going to be doing this each individual. So we were doing these multiple channel tests. Because on these multiple channel tests, we are including about 10 to 12 biochemistry reactions on one channel. So this is what it looks like. This is called a multiple channel tube, enter tube. Now Lee, you're gonna take a picture when I do these actions, because I want to be just save a little bit. Okay. And you can see in this one, including glucose, lysine, sulfide indole, lactose, arabinol, sorbitol, VP, urea, and citrate. So which means each well, they containing a substrate. We can do that certain biochemistry reaction. So by other means, we are doing these individually In here, we do everything together because each wheel containing that specific substrates. Okay, how to do this? So Leah, why don't you come here and I can just do a demo. You will get a one plate which is E. coli. This is actually E. coli 01578H7, so be careful. So what we do it, first thing what you do, come here. First thing what you do is label. Someone can borrow me just drop it down. First thing you want to do is a label. Label on the side. So let's say my name, they're just like CAs. And this is E. coli, we'll just write like that. So just take a picture like that. So what do you do in the label? Okay, well, you can write a date there. So for example, 11, 10, 27, okay. Second, you need to know which one is the head, which one is the tail. So if you don't know, we just take it to look around, okay? See that twist there? That is the end. What you see right here, that is the head, what we need, okay? Needle is the head, that's the tail. So in other words, the white is the tail, blue is the head. So what we do? This is what we're going to do. Using one and just pick one colony right there. Just pick one colony, each individual colony. Okay? Then passing through this. Passing through it. The idea is let all the bacteria going through this channel, but then goes back. Okay? Okay, now right here, goes back until here. You will see a little bit of that niche there. So Lee can take a picture right there. There's a niche there. We don't want any cross-contamination, so we close this side first. Okay, now you have to break this down. Lee, take a picture. We have to break this down. Why this one? I don't think this very well. You should be break this down easy. Wait a second. This one doesn't generate create it very well. We should be create this very easy. Okay, so just take a picture right here. We should break this. Okay. Okay. Then you close right here. Now, what are the things we left? Some of the bacteria they grow aerobically. Some of the bacteria they grow anaerobically. Some of the biochemistry reaction which has happened in the aerobic environments with oxygen, some of them without oxygen. So what we do? You look at the side. There are some of the wheels right here. You have to block it. So Lee will take a picture. We have to block this. Last one is the needle. We have to block it. Okay. Okay. Then this, we will do the incubation, and during the 24-hour incubation, we'll see the biochemistry reaction. Basically, it's a color change. So we'll talk about that next 2 weeks from today. Every biochemistry reaction, according to the results, positive or negative, there is a number for that. Then all the number added together will be a panel number. That panel number is corresponding to one bacteria species. That's what we're going to find out next the 2 weeks from today. Okay? This is a little bit expensive, so what I want to do is 2 of you gonna share. So we're gonna every 2, 2, 2, 2. You can do by yourself, then 2, 2. 1, 2, 3, 4, 5, 6, 7. We're gonna get 7. If some of you who are interested want to do it by you individually, you can just grab another one. I assume in the second and third lab not have many students, so you can, if you want to be interested, you grab one, do individually. This can be your gift from the class. After incubation, it will have different color. As long as you do not open, it's not gonna contaminate it. You can put on your dorm or closet, take it home. Okay, as your gift of this class. And you tell your son or daughter many years later that is multiple channel testing for bacteria called the enter tube 2. Okay, so that's why every time I let the students try to practice one. Okay, so that's what we're going to do today. So what we're going to have, everybody going to come here to get 1 slant for phenylalanine test, semi-solid agar for SIM test, and grab 1 tube from here for Meso Red Vocus Proscia test. All these are very easy, which is you inoculate the bacteria. Now we're going to assign the culture. What we have today is, you guys going to do E. coli. You guys do Pseudomonas aeruginosa, you guys do Proteus vulgaris, and the last group you will do, let me see what we have. Last group you do Staphylococcus aureus. Okay, now I want to make sure one thing. For this guy, for MRVP test, is a little bit different because this is specifically testing enterobacteria and E. coli. So I want you guys, everybody got 2. Everybody gonna go 2. 1 for E. coli, 1 for Enterobacter. Enterobacter is right here, right EA. E. coli is EC. So this one is special. For phenylalanine test is you assign the culture. SIM test is you assign the culture. For this guy, you get 2 tubes, 1 for E. coli, 1 for enterobacteria. Everybody do it individually. Which means 2 weeks from today, you're gonna do 2 set of the test, okay? So everybody gonna get 2. Now, then we're gonna do the enter tube test. The enter tube test is basically 2 of you right here. Basically, 2 of you get 1. If you are interested or very interested, you can do an individual by yourself. Here are the plates. You got to do is those single colony. Here are the plates. Okay? Pick single colony, do what I just did. But be careful, this is O57H7, so make sure you wear gloves and wash hands before you leave here. This is a real foodborne pathogen. Okay? So these are some, you grab a plate, so you do it, then you return here. Okay? So that's what we're going to do. Then 2 weeks from today, we will do identification and urinary tract infection for testing the urine sample. Okay, so that's 2 weeks from today. Okay, so go ahead and do it.